Alrighty, hi guys, it is Jubilee here and I'm back for another TA review. This week we are going to be covering our quiz 5 materials, and so that is going to cover blood and blood vessels, blood and lymph vessels. Alrighty. So first we are going to talk about blood. And so remember when we learned about the 4 basic tissue types, we did say that blood was included in connective tissue. It was kind of like an oddball that kind of just got grouped into connective tissue. So it doesn't necessarily fit everything that you would think of when you think of your normal connective tissue. So it is kind of a specialization of connective tissue. You can kind of think of it as like the fluid form of connective tissue because it is connecting everything together, but in kind of a fluid form, okay? So we're going to talk about the functions of blood. Just know that blood is composed of erythrocytes, so that is your red blood cells. Your red blood cells are erythrocytes. Leukocytes, that's going to be the general term for your white blood cells. And then it's also composed of platelets. We're going to learn in a second those help with clotting. It's one of your big clotting factors. And then they're also suspended in blood plasma. So that's kind of like all the liquid, the fluid that's around there. That's what's going to hold all your nutrients, your proteins, hormones, everything like that within your blood. Okay. And so as you probably already know, the main function of blood is going to be transportation, right? So transportation of those cells and those fluids, those proteins, everything that's in your plasma, in your blood, okay? So your red blood cells are going to carry oxygen to and CO2 away from tissues, right? So make sure you get those directional terms correct because he will switch those up on you, right? Say oxygen from tissues, which would be false. CO2 to tissues, that would be false, right? So just make sure you get those directions right. It seems pretty straightforward, right? But we need more oxygen to our tissues to continue to create ATP, to create energy. And CO2, carbon dioxide, is going to be that waste product that needs to be carried away from tissues in order for them to continue to function. Okay, and then your white blood cells, so your leukocytes, are mainly going to function in immune defense, right? So immune defense, so anything that your body deems as foreign, whether that be a bacteria, some kind of allergen, a pollen, any kind of cell that is not a part of your body or what it recognizes as a part of your body, it will attack. Okay, so that's what your white blood cells do, and they all have different roles in that, but they all do work together for immune defense for your body. And then you've got your plasma. So that's kind of the liquid pool that all of these cells and chunks of cells are sitting in. And so that is where a lot of those proteins are going to be dissolved. So things like glucose, those nutrients, are going to be dissolved in that plasma. Waste from those tissues, so things like urea, right? Urea that ends up in your urine. Things like proteins, stuff like that, hormones from your endocrine system, in informational mediators, so cytokines, any kind of thing like that is going to end up in your plasma, and so that's how it's going to be transported throughout your body. Okay, and then you do also have platelets. So those are those little chunks of cells, basically, and those are going to help with clotting. Okay, so the formation of scabs, the formation of those crusts whenever you start actively bleeding. Okay, so now we're going to learn about the different types of leukocytes, of white blood cells. And you do need to be able to identify all 5 of these different types of white blood cells. You need to be able to describe them to me, to identify them, and you need to be able to tell me kind of their main role or function. Okay, so the first one we're going to start off with is going to be the neutrophil. And so this one is going to be one of the most common types of white blood cells that you'll see very often when you're looking at those blood smears. And so what they are is they're a type of granulocyte, okay? A granulocyte basically just means a cell with granules in it, okay? So it functions by having granules in it. That's how it helps it to do its function. So neutrophils, they're going to be important for bacterial infections and phagocytosis, okay? So they are really good at putting up an immune response to bacteria. What they'll do is they'll actually phagocytize that bacteria. And hopefully you remember what that means, but that's basically where they attach to something and they literally envelop it. They kind of, the way I think of it is like they eat it up. So they envelop it, they wrap their plasma membrane around whatever they're trying to eat. They'll bring it into their cytoplasm. They'll fuse that with a little lysosome, and that's what's going to help them digest whatever they've just eaten. Okay, so your neutrophils are really good at that when it comes to bacteria. Okay, that's what they specialize in, but they're also just good at eating some other random junk as well. Okay, and like I said, they are a type of granulocyte. We'll learn that you have 3 main types of granulocytes within your white blood cells. But the thing about neutrophils is their granules are very small. So you can barely kind of make up some kind of graininess in here, right? But it's not very obvious. And you'll see in some of our other examples, you can barely see that kind of graininess at all. And so that's kind of how I remember them is neutrophils are very neutral. They have a very light cytoplasm. Okay, so the cytoplasm is very light. It's kind of that normal pinkish. And then they do always have this kind of segmented lobulated nucleus. So you'll see here it kind of looks like there's maybe 4 nuclei within this one cell, but you can see that they actually do connect in these very small parts. So basically what that is just that some parts of the nucleus are bigger and more expanded and some parts are condensed, and then it gets big and expands, condenses, and so that's what we call a lobulated nucleus, where there are multiple lobules or these expanded parts within the nucleus, and that is very common in a neutrophil, okay? And so it just depends on the way that this nucleus is sitting, the way that it's orienting itself, how it's gonna look. The nucleus isn't always gonna look the same. They can vary a lot, but it is gonna always be this kind of crazy looking lobulated thing, okay? And the next one we're gonna talk about are lymphocytes. So this is the second most common white blood cell in your body, and these are important for adaptive immunity, right? And so that basically means that these lymphocytes, they will adapt to whatever kind of antigen they see, okay? So this neutrophil, all these other cells, they're going to respond to what they like to respond to. So this neutrophil doesn't really care what kind of bacteria it is, right? It's just going to eat the bacteria. It really doesn't care. With lymphocytes, they are very good at targeting things, but they can only target that one specific thing, okay? So if this lymphocyte is made to target a certain type of bacteria, it will only go after that type of bacteria. If it's made to target a certain type of pollen, an allergen, it will only go after that. And that's just the way that these things are made, okay? This is how we base our idea of vaccines. It's based around the idea of lymphocytes, and that's because if we present your body with this very specific inactivated virus, bacteria, whatever, these lymphocytes are going to look at that, they're going to recognize that, and they're going to build up an immune response for that specific antigen, pathogen, and that specific antigen only. Okay, and so that's how we use vaccines to our advantage. These lymphocytes are very good at doing that, but they can only do it to one specific thing that they're built to defend against, if that makes any sense. Okay, so that's why we call it adaptive immunity, and we'll learn more about that when we learn about the immune response. Okay. And so there are 2 different types, main types of lymphocytes, the B and the T cells in that adaptive immunity. They both do only target that one specific thing that they're built for, but they target them in kind of different ways. I won't go into too much detail with that. And then as far as their appearance, they are the smallest white blood cell. Hopefully you can appreciate that. They're only a little bit larger than these red blood cells. right? They're only a little bit larger than the red blood cells around them, and they have a very large spherical nucleus, okay? And so you can see here, this whole lymphocyte is pretty much all that kind of dark blue, right? And it is all that color because that is all the nucleus of this lymphocyte. So this lymphocyte is almost entirely nucleus. You can see a little bit right here of just the cytoplasm, right? So there's just a little bit of cytoplasm around the nucleus in this lymphocyte, but it is almost entirely nucleus. And that is how I know that it's a lymphocyte. Okay, they're also very small, they're very perfectly shaped, they're very round and perfect looking, and that's how we're going to identify our lymphocytes. The next one, so this is where they start to get a little more rare, a little harder to find, are going to be monocytes. And so this is going to be our monocyte, and what they are is they are our phagocytic cells, okay? So they are the precursor to macrophages, and macrophages are our phagocytic cells. So if the monocyte is floating around in the blood and it gets some kind of message like, hey, something's going on over here, there's damage over here, there's stress signals coming from the tissue over here, it's going to pick up on that and it's going to embed itself into the tissue. That's when it becomes a macrophage and is no longer a monocyte. Okay, so monocytes are in the blood, macrophages are in the tissue. What that macrophage is going to do is it's going to phagocytize things like I described earlier in the neutrophil. It's going to basically wrap its cell membrane around it and eat it, digest that material. And they are really good at that. They also don't really care what they eat, okay? If the body tells it to eat it, if it seems like something that's foreign, it's going to eat it, it's going to digest it. It also is an antigen presenting cell, so it's going to eat that thing and it's going to save a little piece of it and present it to these lymphocytes. Okay, I don't want to get into too much detail about this because, this is a lot of immunology if I get into too much detail. We mainly just want to know the big kind of role. So phagocytosis, precursor to macrophages in these cells, and then how to identify them. So these are the largest white blood cells. A lot of people will say they have a horseshoe-shaped nucleus. So this doesn't necessarily look like a horseshoe shape, but basically what it means is it's this kind of large nucleus and it does kind of have this more prominent dent on one side, right? So this kind of prominent dent. So it's kind of like a U shape, a soft U. And of course that will vary between each cell. So they're not always going to look perfect like that, but they are very large. They have this kind of large dented nucleus, and then they will have this very kind of grainy blue-gray cytoplasm. Okay, so you can see how the cytoplasm of this cell is different than all the other white blood cells. Okay, and then you're going to have your eosinophils, so that's even more rare. Eosinophils are important in parasitic infections. Okay, that's what we mainly think of when we think about eosinophils is parasitic infections. They are another granulocyte. So hopefully you remember our last granulocyte was the neutrophil. So that's another granulocyte. So they've got these big red granules in them. So you can see all these big red kind of dots. Those are those granules in this granulocyte in this eosinophil. Okay, and they do help fight off parasitic infections. And then they are also lobulated cells, so they have this kind of lobulated segmented nucleus. So it looks like there's two nuclei in here, but you can see it's connected, right? So that is a lobulated nucleus, and they are normally bilobed, meaning that there's two big lobes in here. Neutrophils normally will have more than that. Eosinophils will normally have two. Okay. And then the most rare one that you really don't expect to see any of in blood, you wouldn't think it's abnormal if you didn't see any, would be basophils. These are important in the inflammatory response. So just like our mast cells that we learned about, they help with releasing histamine. Histamine is an inflammatory mediator. So what it does is if they find something going on, they'll release that histamine and that calls other cells to the site. Okay. And so another thing that they're associated with, so they're associated with inflammation, right? So calling other cells to the site, but they're also associated with allergies, which is kind of the same thing that happens is they call all these other cells to the site, but they're doing it because they send something that's not actually bad for your body. So something like pollen, right? Something like grass that's not really going to cause harm to your body. Your body just senses it as bad, so they kind of just preemptively release histamine and cause all this inflammation when they shouldn't really be. So they are kind of associated with allergies as well. Okay, they're going to be our third and final granulocyte. So all of these, the eosinophil, the neutrophil, and the basophil, are going to be our granulocytes. So all the ones with the phil kind of ending, if that helps you. And so their granules are going to be these very big kind of dark blue granules. Okay, so these very big dark blue granules that are kind of all throughout this basophil, that's how you're going to identify it. It does have a lobulated nucleus. It's just you can't really see it because the nucleus is going to be kind of this purpley blue, right? And the granules are kind of a purpley blue, so it kind of gets in the way, right? But yeah, so these are our granulocytes. They also all have lobulated nuclei. Let's see if I can go. All right, so now we're going to do some practice. So here you can see in this one, right, we've got a whole bunch of red blood cells, right? We've got some little platelets right here. These are those little chunks of cells that help with clotting, a bunch of red blood cells. And here you can see we have some leukocytes or some white blood cells, right? So we're going to try to identify these. Here's a zoom in of them, right? So hopefully you can see this one on the left. It kind of has this light neutral cytoplasm, right? There's kind of a crazy thing going on with the nucleus. So we might think it's maybe segmented, it's lobulated, and that's kind of doing whatever it wants. So hopefully you realize this is a neutrophil. This is a neutrophil, right? It's got that very characteristic light pink cytoplasm, light pink neutral cytoplasm, and kind of a crazy looking lobulated nucleus. This one right here, hopefully you can appreciate how the cytoplasm is much more red, right? This cell is actually more red than the red blood cells around it. Hopefully you can appreciate that. And that is because of those very big red granules in this cell. So hopefully you realize this is an eosinophil. Eosinophil. Okay. And here's another trick to help me remember this is, remember when we learned about the stains, we said that eosin stains proteins red, right? Eosin is the part of H&E that stains proteins red. So eosinophil means eosin loving. So it loves the stain eosin, so it's going to stain very red. Okay. But yeah. And then hopefully you can appreciate that it kind of looks like there's two nuclei in here, right? But really we know that that means that it's bilobed, and eosinophils are very commonly bilobed. Okay. If we look at these really quickly, hopefully you can realize how all of these are the same kind of white blood cell. They all look very similar, right? Their nuclei are all kind of doing different crazy things, but they do all look segmented. They do have very neutral light pink cytoplasm. So these are all going to be neutrophils, okay? Hopefully we can look at these ones and see how in here it looks like the cell is almost entirely that blue kind of nucleus color, right? And so it looks like the cell is just kind of this one constant shade of that dark blue. So this cell is almost entirely made up of its nucleus, right? It's very small. It's smaller than a red blood cell, and it's very uniform. So that's going to be a lymphocyte, okay? This lymphocyte's a little larger than normal, but yeah, we won't get into too much detail on that. And then this one right here, hopefully you can appreciate, it's not the same as a lymphocyte where almost the whole thing is nucleus, right? And it is quite a bit larger. It's quite a bit larger. And then another thing is it has this kind of bluish, it's a little more blue cytoplasm. There is kind of a dent if you use your imagination on the nucleus. This one we're going to call a monocyte. Okay. All right, so now we're going to do some more practice. So here we go. So these ones right here, you see how they have very light pink cytoplasm and crazy looking nuclei? Those are going to be your neutrophils, okay? This one right here, same as this one, you can see the cytoplasm in this one is really red, right? Much more red than the red blood cells. And then we do kind of have this bilobed nucleus going on. So that's an eosinophil. This one right here, I can see how you might not be as sure about this one. You could go with either a lymphocyte, because it is this kind of big blue blob, or you could go with a basophil, right? And I'm going to tell you right now, this is a basophil. And the reason I know that is because the blue that's in here is very grainy and patchy and irregular, right? And so that is due to the actual granules in the cytoplasm, not due to the nucleus. If it was due to the nucleus in a lymphocyte, the blue would all be very similar in color and texture, so it wouldn't have this kind of graininess that a basophil does. Okay, so that one we're going to call a basophil. Okay, so this is just talking about margination of white blood cells. So when we talk about white blood cells marginating, that's basically how they're going to exit the blood vessel to enter the tissue, right? Because if I have a cut or something and there's bacteria in that cut, those bacteria are going to be in the tissues, right? They're not going to go straight into the blood, or at least I hope not. So they're going to be mainly in the tissues, and that's mainly where we have to fight this off. And these white blood cells need to have a way to get into the tissue and start phagocytizing things and start trying to fight off this infection. Okay, so what happens is if they get signals, right, they get like histamine, they get little signals that something bad's going on here from the tissues, there's necrosis or something, what's going to happen is they're going to marginate. So they're going to line up on the side of this blood vessel. So you see how they're sticking to the side of this blood vessel, and they're actually going to squeeze through the endothelium and off into the tissues where they can start fighting things off. So here you can see this is an eosinophil, but it is not in a blood vessel, it's actually in the tissues. Okay, so that's called margination. So yeah, and then there are also some plasma cells that you can see in here. So we'll say that this is maybe a plasma cell. And we'll say maybe that's a plasma cell. Let's see, that one could be a plasma cell. So plasma cells are going to be those cells that look like a cracked egg. Okay, cracked egg. So they have about as much cytoplasm as they do nucleus. And what they are is that's what a B cell turns into. So a B cell is a type of lymphocyte, and they will turn into plasma cells. Plasma cells will sit there in the tissue and produce antibodies, right? Antibodies to try to fight off and attach to that antigen. Okay, so now we're going to go over EMs of these white blood cells. And they can be very tricky. So I know when I took this class, I did not know how to tell the difference between all the different white blood cells. I just kind of guessed. And that's because nobody taught me how to tell the difference. So we're going to kind of go over that. And hopefully once you guys learn these little tricks, you'll realize it's actually not that bad and you don't really just have to guess. You can actually figure it out. Okay, so the first one we're going to go over. So this one right here is an eosinophil. Okay, and a little disclaimer, don't try to compare how it looks in light microscopy to how it looks in electron microscopy, because it's very different, right? In light microscopy, we've got colors that stain for certain things. In EMs, it's completely different, okay, the kinds of things that we're going to pick up. So this is an eosinophil, and the way I know it's an eosinophil is that on EMs, on electron microscopy, eosinophils have this thing called a crystalline core within their granules, okay? So hopefully you can appreciate this is the nucleus of that eosinophil doing whatever it feels like, and all of these are those little granules, right, that on light microscopy look red, those red granules, right? And hopefully you can appreciate how these granules, within this one granule, I have two different colors or textures, right? Within this one granule, I have two different colors or textures, right? What that's called is a crystalline core, okay? So this is a really good example. This granule right here, it's a crystalline core within that granule, okay? This one's a good example right here. And that is characteristic of eosinophils, okay? The next one is going to be a neutrophil. So here's a neutrophil. Hopefully you can appreciate this is the nucleus of that neutrophil. This is all the cytoplasm with all the little granules going on. And remember we said in the neutrophil they have very small granules, right? You can't really see them on light microscopy. And you can see how these granules are much smaller than these ones in the eosinophil, right? And so what a lot of people will say is they have like dumbbell shaped granules. right? It depends on how you're looking at it, but you could maybe say that that was a dumbbell-shaped granule. So it's just these very small granules throughout the neutrophil. Okay, and let's see. And so now we're going to look at a basophil. Remember, this is our last type of granulocyte. And hopefully you can appreciate how very different this one looks, right? So these are very large uniform granules, right? So you can see how within one granule, it's all the same kind of color and texture, right? That's very different from our eosinophil, right? So you see how that's different. And then they are very large. They take up a lot of the cytoplasm, these little granules in the basophil. Okay, so hopefully you can appreciate how that's different. Next is going to be a lymphocyte. And the way I identify as a lymphocyte is it is mostly nucleus, right? Just like in light microscopy. So this is a lot of nucleus within the cytoplasm of this cell, right? There's not very much cytoplasm. They are also very pretty, right? So this is such a cute little perfect circle, just like it is in light microscopy. So it is very pretty, relatively circular, relatively small, and it is mainly nucleus. Okay. And then the next one is going to be a monocyte, and this is the one that people kind of have trouble with the most. This one in the neutrophils. But the way I'll kind of tell you is, remember we said monocytes are important for phagocytosis, right? They turn into macrophages, and macrophages phagocytose everything, right? And I said when you are doing that, so they're basically taking their little cell membrane, they're wrapping it around something, they're giving it a little hug, and then they're enveloping it in their cytoplasm, okay? And so they're going to eat it up and digest it and whatever. And so what I tell people is don't really look in the cytoplasm. That doesn't help me. You can look for these autophagic vacuoles or whatever, but it depends on what the macrophage or the monocyte is doing at the time. The way I identify them is I look at the border of this cell, and you can see how the border of the cell has all of these plasma membrane projections, right? You can see all these. So it's kind of like little arms branching out on the plasma membrane of the cell, and that is how it grabs a hold of all of these little pathogens and germs and whatever. It grabs a hold of them and eats them up. Okay, so it needs these little arm-like projections, and so that's what I look at when I look for a monocyte, are these arm-like projections. Okay, so now we're going to do some practice. So let's see what this one is here. So hopefully you're thinking maybe this is a lymphocyte, because the nucleus is very large, right? It's very pretty and circular. There's not very much cytoplasm, and it is relatively small, okay? And then I know a lot of you guys will get confused and you'll say, well, what is this arm-like projection? What is this arm-like projection? Maybe this is a monocyte, right? And I'll tell you, not every cell, the cells aren't going to look completely perfect, but hopefully you can appreciate how this little kind of thing, this little thing is different than this cell, okay? So this cell, do you see how big this projection is and how big this projection is and how far it sticks off of the cell? This is a monocyte, okay? So this is that big arm-like projection that I'm talking about, okay? And then this is actually grabbing something and it's about to bring it in. Okay, so this is a monocyte. We'll do some more practice of this one right here. It's tricky, right? So this is not a basophil. It doesn't have those large, similar granules. It's not an eosinophil. It doesn't have those large crystalline core granules. It's not a monocyte because we don't really see very many arm-like projections. It's not a lymphocyte because its nucleus is relatively small compared to its cytoplasm, right? And then we do have all these crazy looking maybe granule things. So this is a neutrophil. Okay, this is a neutrophil. This is a neutrophil. This is a neutrophil. This is a neutrophil, right? So a lot of these ones, if it doesn't fit in that into one of those specific categories that you know, it's going to be a neutrophil. Okay, and remember we did say that those are the most common. Okay, but something like this, you see how this is different? This one looks really pretty and perfect and circular. It's mainly nucleus. This is going to be a lymphocyte, right? This one right here, a lymphocyte. This one up here, a lymphocyte, right? And then this one right here is a monocyte. It's actually the same one as this one right here, but here you can see those arm-like projections. Okay, so hopefully that helps you guys. Now we're going to talk about blood cell formation. You know, there's a lot of different stages in this and they all look very similar and it does confuse people. So I don't want you to get too bogged down in the details. You can just kind of use Dr. Johnson's slides for the most part for the quizzes. And as far as the midterm goes, I won't be too picky on that for you guys, I promise. But just know that all cells, all your red blood cells, white blood cells, everything like that are formed in the bone marrow, or they all come from common precursors in the bone marrow. There's a few exceptions and stuff, but we won't get too bogged down in that either. And they do all look relatively the same before they start to differentiate and look like their end goal cells. Okay. And hopefully you can notice some trends here. So right, they all start out with blasts. Then we've got some like something-ocytes, then we've got myelocytes, then we've got metamyelocytes, then we've got bands, and then we've got the fully formed cell. All right, so they all kind of go through sort of the same process to become the end goal cell. Some things I do need you to know is that lymphocytes, they make T and B cells, right? And so those B cells may differentiate into plasma cells. So that's what we talked about earlier. They go into the tissues, they turn into plasma cells, and they make antibodies, right? Another thing that I need you guys to know is monocytes will differentiate into macrophages in the tissue, right? So we've already kind of covered that as well. And then another thing I need you guys to know is that platelets come from megakaryocytes. Okay, megakaryocytes. And we'll look at that in just a second, how that works, but basically they're just breaking off little chunks of themselves, and that's kind of making a little platelet chunk. Okay, so just kind of know the general progression of differentiation from these. So they all kind of start out as the same kind of generic looking cell, and the more differentiated that they get, the smaller and more condensed their nucleus is going to get. Okay, and it's kind of just because when they're big, everything in their nucleus is kind of open to possibilities. They don't really know what they're going to do. Once they know what they're going to do, they'll start closing down compartments that they know they don't need, right? That they know they don't need. So the nucleus becomes more condensed, and it starts to look more like what it's supposed to be. Okay, so this is talking about erythropoiesis, specifically, just to go over some of like the kind of naming conventions, right? So we've got the pro-erythroblasts, and these are the most kind of generic looking ones, right? You can't really tell what it's going to be. The nucleus is huge. Then we've got basophilic erythroblasts. It looks pretty much the same, right? It's just the nucleus is a little bit fractured. It's kind of starting to break down a little bit. And then the cytoplasm is yeah, kind of looks the same, sorry. Then we got the polychromatophilic erythroblasts. So the nucleus is condensing even more. You can see there are little areas where the nucleus is darker than others, so that's what the polychromatophilic means. It means more than one color, loving, so it's going to stain with kind of different colors and textures within the nucleus because it's condensing in different places. Then we've got orthochromatophilic erythroblasts. So this is where it starts to get more mature and starts looking like it's actually doing something. So the nucleus is very condensed. It's starting to get much more mature. Then it will actually lose its nucleus. So it will lose its nucleus and then it becomes a reticulocyte. Okay, so this would be a reticulocyte. This is what we call an immature red blood cell. A mature one, remember, is an erythrocyte. So reticulocytes will actually be released into the blood, and they have to go through several little rotations in the blood before they can become erythrocytes. Okay, so reticulocytes don't have a nucleus anymore, but they're a little bit more blue and gray. You can see how this one's a little bit more blue and gray, purpley than this one right here. So this one's an erythrocyte, this one's a reticulocyte. The reason is the reticulocyte still is making hemoglobin, right? It still needs to make more hemoglobin to be a fully mature erythrocyte. Remember, hemoglobin is the main protein in erythrocytes. And so in order to make that hemoglobin, it still needs mitochondria and it still needs ribosomes and everything like that to make those proteins. And so that higher concentration of mitochondria, of ribosomes, of everything like that makes it stain more blue. And then in the erythrocyte, once it's fully mature, it is chocked full of hemoglobin, it is chocked full of protein, so it's going to stain much more red, much more pink. Okay. So now we're going to talk about megakaryocytes, and remember we said they form platelets, and they are the largest cell in the bone marrow. So here's an image of the bone marrow, and hopefully you can see and appreciate that this is one cell and it is much bigger than all the tinier cells around it, right? So this one large cell is a megakaryocyte. Here's another one, a megakaryocyte. And so what happens is you have this really large cell and it actually breaks off pieces of itself, little chunks, and that's what makes a platelet. So here you can see this is one big megakaryocyte, and you can see how it's forming all these little kind of fractures and cracks out of vacuoles, and it's going to break off those chunks, and that's going to form a platelet. Remember we said monocytes form macrophages, so those macrophages are going to go into the tissues and eat stuff. So this is in the lungs, and so hopefully you can appreciate these are all macrophages that have eaten up carbon or tar or junk or something, and they're trying to digest it. That's why these granules are all kind of different colors, because they're at different stages of digestion. So these are macrophages that came from monocytes in the blood. Okay, and here we're talking more about those reticulocytes versus erythrocytes. So this is that same picture. Hopefully you can appreciate this is much more purple. This is much more red. Hopefully you can appreciate how there's a little hint of blue purpley in this cell versus these cells behind it, right? That blue purpley color. So those are reticulocytes. They're immature erythrocytes. And here you can see in an EM that reticulocyte still has mitochondria. It still has little ribosomes spotted throughout here. This erythrocyte, totally protein. A whole bunch of hemoglobin. That's basically the only thing that's going on in here is hemoglobin. Right, so hopefully you can appreciate how that's different. Okay, so now we're going to talk about vessels. I'll try to make this kind of quick because it is kind of straightforward. So blood and lymph vessels, their functions are pretty straightforward. So because we are multicellular, right, I have to deliver nutrients all the way from your stomach to your brain, vice versa, everything like that. So there's a lot going on. So that's why we need lymph vessels, is to transport all that blood from one place to another. So they help to distribute oxygen, nutrients, and hormones, as well as collect waste and transport that waste to your organs like your kidneys and stuff like that, where those organs can eliminate that waste. Okay. So this is that diagram that kind of talks about how your blood vessels progress throughout your system. And it can get kind of complicated, so I'll try to dumb it down a little bit. But basically how it works is your heart is a blood pumping machine, right? And so the way it works is your heart is on and then it's off. It's on and then it's off. So it creates this enormous amount of pressure, 120 milligrams of mercury, or millimeters of mercury. And so it goes from 120 to 0 as it relaxes, 120 to 0 as it relaxes, right? So that is a very big difference in pressure, right? And as it's doing that, so it's going to, if that went to your tissues, right, just like that, if it went 120 milligrams mercury to 0, that would be crazy to your tissues. It would stress them out. They would not be able to deal with that because they're getting all of that oxygen, all of that nutrients all at once at a crazy high pressure and speed, and then they get nothing. And then they get all of it at once, and then they get nothing, right? So your tissues cannot deal with that. But your heart needs to pump it like that for it to get all the way to your toes and your fingertips, right? And so the way we slowly change that pressure to make it constant to where your tissues can deal with it is through the system of your blood vessels, okay? So we're going 120 to 0 in your heart, and what happens in your aorta and your big arteries is they're very elastic, so they don't let it drop to 0. Okay, so when it wants to drop to 0, your arteries will actually squeeze down because they're elastic to maintain that pressure. Okay, so they conduct blood to maintain pressure during diastole, during that rest period. So pressure does not drop to 0 like it does in your heart. Okay, so that's what your arteries and your aorta do. So they keep it at this 120/80, 120/80, right? And hopefully you realize that's what blood pressure is if you have a normal blood pressure, you have 120/80. That's measuring your arterial blood pressure, right? And then we still need it to drop even further, right? So in your arterioles, that's where they provide resistance. They provide peripheral resistance to that blood flow so that this kind of pulse that you feel, 120/80, 120/80, in your arteries gets smaller and smaller and smaller and smaller until there is no pulse at all, until it's the same no matter what your heart is doing. So that's what your arterioles do, is they bring this pulse down to nothing. So there's not a pulse, there's not a big difference, right? Depending on what your heart's doing. And that's when we get to the capillaries, because in the capillaries, we do not want there to be a pulse, right? Because the capillaries is where we're exchanging nutrients to the tissues. And remember, we said the tissues cannot deal with a pulse like this. So in the capillaries, we don't have a pulse. And then pressure keeps dropping and dropping and dropping till we get to our veins. It gets very, very low, and it actually gets a little bit negative in the vena cava, the largest vein in your body. Okay, and that's how this negative pressure will feed back into the pressure of 0 in your heart and start all over again, 120/0, 120/0. Okay, so hopefully that makes sense. This is just saying how your vessels actually maintain a constant supply of blood to this very important part, your capillaries, where we're actually exchanging that nutrients. Okay, so now we're going to talk about arteries versus veins. I know you guys kind of struggle with identifying the difference. And so remember we said arteries have to deal with that big change of 120/80, 120/80, right? And so in order to do that, they need to be more stretchy, they need to be more sturdy, and so they have a little bit extra stuff going on, okay? So both of these two kinds of vessels have the same 3 main layers. So a tunica intima, that's the innermost layer, okay? They both have a tunica intima that's going to be made of the endothelium, the subendothelial layer, and in arteries, it's going to have an internal elastic lamina. Because remember I said these arteries are elastic so they can recoil to maintain that pressure, so that pressure doesn't drop to nothing, drop to 0 when the heart relaxes. So it's kind of like a little rubber band springing back. Okay, we don't need that in the veins. And then they both have a tunica media, so that's got a bunch of connective tissue and smooth muscle in it as well. But in your arteries, you have a lot more smooth muscle, quite a bit of smooth muscle. You have elastic fibers and an external elastic lamina. So more elastic stuff to help with the kind of recoil of this artery. And then we have a tunica adventitia for both of these. So that's basically the connective tissue that anchors this vessel down to the surrounding tissues so it doesn't move anywhere. Okay, so both of them have these 3 main layers. Tunica intima, so tunica intima would be right here, would be that innermost layer of this vein. Tunica media is right here, right? Tunica media on this vein would be right here. Tunica adventitia is the connective tissue that anchors it down to the tissues around it, so that outermost part, okay? And then the artery has kind of extra stuff, right? And then the way that kind of helps me differentiate between the two is arteries have much more smooth muscle in them compared to their lumen size, okay? So that smooth muscle helps to create kind of a regularity in these arteries, and it holds them open. So here you can see this one, you can see how thick that kind of smooth muscle layer in the tunica media is, and you can see how this artery is smaller than this vein, right? their lumen size. This artery's lumen is much smaller than this vein, and yet this artery has more smooth muscle than this vein. Does that make sense? This one's smaller but has more stuff supporting it than this one that doesn't have very much stuff supporting it. And that's typical of arteries versus veins. So veins won't have as much stuff supporting it as you'd expect for their size. Arteries have way more supporting it than you'd expect for their size, for their lumen size. Okay. So here's some examples. So here you can really compare this, right? So this is a zoom in on this one right here. And then this is a zoom in on the aorta. And so this one is a really big vein. This is, I think, the hepatic vein. So it's one of the biggest veins in your body. And so you can see how this is the inside of the vein. And here you can see there's that tunica media. So there is some smooth muscle here, right? There is some smooth muscle here. And this is the aorta. Do you see how big this tunica media is? It is so much thicker. And these are relatively about the same size lumen-wise. Okay, but this artery has way more support on it, and that's because it has to deal with that pulse that a vein does not. There is no pulse in your veins, but there is a large pulse in your arteries. Okay, and then you can also see there's elastic fibers. So these kind of swirly things mixed in throughout the smooth muscle, those are those elastic fibers. And again, that helps with kind of the springiness and the stretchiness of that artery. So here's more examples. So you can see here, let's look at this one right here. So you can see it's very circular. It's got a very thick kind of lining around it. So this is an artery or arteriole, right? Same with this one, very thick lining around it. Okay. And then this one right here, you can see there's not a very thick lining around it, right? Compared to this one, very thick lining. So this is an artery, this is a vein, okay? And then another way that you can tell the difference is because these arteries have a very thick lining of that tunica media around it of smooth muscle, it keeps them relatively circular, right? And that's important because we want our arteries to always stay open and to stay perfectly circular. Veins, we don't really care if they collapse. They don't need that kind of supportive network or whatever. We don't care if they collapse. And so they're not always going to be perfectly circular. Most of the time, they're going to be very squished and weirdly shaped. So here you can see this, I know, is a little vein or venule because it's squished and it's not very circular. This one is kept open and very circular, so I'd say maybe this is an arteriole. Okay, so hopefully that helps you all keep the difference. So now we're going to talk about capillaries. Capillaries, remember, this is where that exchange is actually taking place of oxygen, of CO2, of nutrients, of hormones. That's where the exchange to the tissues and the blood are actually taking place. And so there are different types of capillaries depending on how much exchange we want to actually happen. Okay, so we're going to learn about three main types. So that's going to be continuous, fenestrated, and sinusoidal. And so these just kind of change depending on how much actual exchange we want going on between the blood and the tissues. So these are going to be our three main types I just talked about. So continuous capillaries is just like what it sounds like. So it has a complete basal lamina, and it is the one that is most protective. Okay, so it allows the least amount of stuff to come from the blood into the tissues or vice versa. Okay, so it allows them to talk to each other the least. Okay, and so that's going to be in important places like your muscle, your testes, your brain, and the thymus. Okay, and so remember, hopefully you guys have heard before, like the blood-brain barrier, the blood-thymus barrier, blood-testis barrier, blood-muscle barrier. And that's because it is this special kind of barrier that doesn't allow certain things through, right? So there are certain things like certain chemicals, drugs, anything like that, that will not go through to your brain because your brain doesn't want to come in contact with them. It's going to let all the other organs deal with that, right? Same with your thymus, your testes, your muscle. So it's a special kind of capillary with a complete basal lamina, and it is called continuous capillaries. Okay. Fenestrative capillaries are where it'll let some things through. Okay, so fenestrated, it'll let some things through. It does still have a complete basal lamina, but the endothelium around it leaves little gaps. Okay, so that's going to be in places like your glomerulus in your kidneys, and in your adrenal gland. Okay, so if you think about, for example, the role of your kidneys, it's to filter things, right? Your kidneys filter your blood and they excrete out waste, right? And so we want to allow that waste, we want to allow things in your blood to exit that blood and end up in your kidneys and eventually in your bladder. Okay, so that's why we have fenestrated capillaries. We don't want to let everything through, whole blood through, right? You don't want to pee out blood, but we want to let a good amount of things through, and so that's why we have fenestrated capillaries. Okay, the next one is going to be discontinuous or sinusoidal, and this is where there are giant gaping holes in your capillaries. Okay, so the basal lamina is incomplete or lacking, and there are giant gaping holes in your capillaries, and so this is going to let whole blood through. Okay, so this is what's going to let a lot of things through, and so that's going to be in places like your liver, your spleen, and your bone marrow, because if you think about what your liver and your spleen do is they filter whole blood. Okay, and so they need large amounts of blood to go into the tissue so that they can filter it, so that they can figure out what's going on, fix it, add things, take things out. Okay, so that's how they function. Your bone marrow is discontinuous or sinusoidal, because remember, we're making blood cells, so we need those blood cells to be able to leave the bone marrow and get into the blood. So that's discontinuous capillaries. So now we're going to look at just a quick couple examples of lymph vessels versus veins. So remember, your lymph vessels carry lymph, right? There is no pulse in your lymph vessels, right? So they don't have to deal with resisting that pulse like your arteries do. So just like your veins, they're going to have valves. They're going to have those one-way valves. And they're not going to have a very big tunica media. They're not going to have all those extra layers. They're not going to have a ton of smooth muscle around them. And so they can look very similar to your veins, because they are these kind of floppy looking things with very thin linings. Okay, so the main difference is whenever you make a slide of something in histology, you drain out all the blood, but you don't drain out all the lymph. Okay, so when you look at lymph vessels versus veins, lymph vessels will still have that lymph within them, so it's this kind of pink staining liquid or fluid. Your veins will be empty, okay, so you've got white inside of them. So that's how I know that this is a blood vessel, not a lymph vessel. Okay, they are both going to have valves. So here you can see there's a valve in a lymph vessel, here's a valve in a vein, right? But we've got this kind of pinky stuff. Here's some more pinky stuff. So these are lymph vessels. There's one with pink in it. That's a lymph vessel, right? This one would be a venule lymph vessel. Okay, so now we're going to do some practice. So which blood cell / bone marrow cell / tissue cell is derived from the type of cells in the top electron micrograph? So we're going to identify what's in this top electron micrograph. And so hopefully you can realize we've got these long arm-like projections, right, on the outside. So this is going to be a monocyte, right? This is a monocyte. And so what did we say is derived from a monocyte? We said it was a macrophage, right? Macrophages come from monocytes. So hopefully you can tell this is a macrophage in the lungs that we talked about earlier. This one, he says largest cell in the bone marrow. Remember, we said the largest cell in the bone marrow is going to be a megakaryocyte. Megakaryocytes make platelets. They don't have anything to do with monocytes or macrophages, so that's not B. And then the smallest cell in the blood, and then here you can see this really large nucleus, and it's very spherical and cute and perfect. This is going to be a lymphocyte. So again, that has nothing to do with monocytes right here. So the only one that would match would be A. Okay, here, which cells best match their image and description? So A, neutrophil, a horseshoe-shaped nucleus. So this is a cell with a horseshoe-shaped nucleus, right? It's got that little indentation in it on one side. That's kind of this soft U shape, but that is not how we would describe a neutrophil, right? That's how we would describe a monocyte. So that one is wrong. Those do not match. B, eosinophil. So this is an eosinophil. It's got those bright red granules, And then it says lobulated nucleus. And yes, we did say eosinophils have that lobulated nucleus, right? A bilobed nucleus, more specifically. But yeah, a lobulated nucleus, so that one does match. C, a basophil, and it says large spherical nucleus. So this is a basophil. We can see those kind of big, thick blue granules in here. But when we describe a basophil, would we describe it as having a large spherical nucleus? No. Remember, we said basophils also have a lobulated nucleus. You just can't see it because it's behind all of these granules. We would describe a lymphocyte as having a large spherical nucleus. So those do not match. Okay, so it's trying to be tricky in this one. Everything, all three things, so the name, the description, and the picture all have to match. So that would only be B. Okay, this one is talking about that big old complicated diagram. So this illustrates the blood pressures and luminal surface areas of different blood vessels. Which blood vessels match? So here, so we've got these different little hash marks. We can go through and identify all of them. So remember we said right here at this is where we're going 120, 80, 120, 80, 120, 80. So this would kind of be where your aorta is, right? Remember your heart goes 120, 0, then it goes straight into your aorta, and that kind of sets it up for 120, 80. So that'd be your aorta. A, so we still have this really big pulse. So this would be your big arteries. So this is still your arteries. Okay, so we still have this big bounding pulse in your arteries. B, so this is where we're really starting to get rid of that pulse, right? We're really starting to, we start with a big pulse and then we whittle it down to nothing. Remember we said the type of vessel that's responsible for that is going to be your arterioles, B. So that's peripheral resistance right there. Here, remember, this is where we said it's the first place where we don't have a pulse. It's the greatest surface area because that's where all your tissue exchange is happening. This would be indicating your capillaries, this area right here. Okay, and then here would be where the pressure keeps going down and down and down almost to 0. That's where you have your veins, right, as the pressure drops and drops. And then as it gets negative, that would be your vena cava going back to your heart. So if we match that with this one, A, arterioles, no. This would be your arteries, right? B, capillaries, no. This would be your arterioles, right? This is your capillaries. And then C, veins, yes, that would match. So only C would match. Hopefully that makes sense to you guys. Okay, which structure is / are blood vessels, right? And he underlined blood vessels. So A, we've got this very thick lining around it. It's very circular. So I'd say this is a little artery or arteriole. So that is a blood vessel. Okay, so A is one. B, remember we've already learned about this type of tissue. This is adipose tissue, right? It looks like chicken wire. So this is not a blood vessel. This is adipose tissue. This is white fat. C, it does kind of look like a vessel. We've even got a little valve in here kind of floating through the middle that got cut off. But remember, it's filled with this kind of fluid. So that would be a lymph vessel. It's still filled with lymph. So it's not a blood vessel. It's a lymph vessel. So it would only be A. Okay. And yep, so that's the end. Hopefully this was helpful for you guys. And good luck on your quiz.